Thank you, Robin, for the kind introduction. I would like to thank the chILD Foundation for the invitation. This is an audience I'm not used to talk to, so I'm a little bit nervous, but also happy. And I hope in the next 15 minutes I will convince you that our lines of investigation can be part of the future of child research. So as you can see, I work at the Pulmonary Center. I work with Dr. Darrell Cotton, and also our research teams are part of the Center of Regenerative Medicine of Boston University. So if I was to state just one slide what the major goals of our research are, is first of all to understand and model lung developmental disease, and also to develop new drugs or cell-based therapies for lung epithelial reconstitution. So we think that the way to do that is to combine data from different systems and combine in vitro and in vivo studies. So we want to get information from developmental biology, and developmental biology is simply the studies of the organisms, how they develop, for example, from the moment that we have the fertilized egg to the moment we have a newborn. And also using the in vitro systems, embryonic stem cells and induced pluripotent stem cells. In the next couple of slides, I will explain what these systems are. So these studies can inform each other and hopefully can derive information that will help us to do disease modeling in vitro, drug development, and eventually derive functional and transplantable lung epithelial cells. So for those who are not familiar with embryonic stem cells, I think it's a good introduction from, I took this cartoon from NIH, a book that explains the notion of embryonic stem cells. So an embryonic stem cell is in fact an artifact of cell culture. What we do is we take cells from the blastocyst stage of development, and we put these cells in culture when we then can expand them. Essentially what we do, we take cells from a certain developmental stage and we freeze them at that developmental stage. Because in development that doesn't happen, everything is transient. Cells move around, they change identities, they form tissues and organs, but they are not fixed. So why these cells are so important and why they can be in use in vitro studies. They have two characteristics. One is that they can self-renew in culture, so they can give copies of themselves. And the other one is what we call the property we call pluripotency. Pluripotency means that they can give rise to all somatic cell types. And that makes sense because when one look at the blastocyst, this mass of cells will give rise to the whole organism. So they maintain this property also in vitro, and they can give rise theoretically into all cell types. And that's important because then we can use these cells to model development and also to derive cells that can be used for cell therapies. But there are some major limitations for this system that don't allow us to use extensively, especially the human embryonic stem cells. So one is a technical hurdle. It's kind of difficult to derive embryonic stem cells, mostly human embryonic stem cells. The other one is the ethical concerns. There's been a debate raging for years if we can use or not human embryonic stem cells because they're derived from a human embryo. So they entail the destruction of a human embryo. And also it's the immunogenicity issue. So as any cell, if we try to use them for cell therapies, they don't come from the same person, the same organism because they are embryonic, and we can have immunorejection issues. So the way to address these issues came about 6 years ago with the development of the induced pluripotent stem cell system and what this is. So 6 years ago, a group in Japan, headed by Yamanaka, discovered that we can take somatic cells, for example, differentiated skin fibroblasts, then infect them with viruses that were encoding for transcription factors. And transcription factors are proteins that bind DNA, DNA binding proteins, and they're very important because they can control, by definition, the expression of a lot of genes. So we saw that by infecting these cells, overexpressing 4 transcription factors in somatic cells, we could derive a cell that was a pluripotent cell. And that was a major discovery. That's been something that has been, this idea has been around for several decades, but it was only in 2006 that became reality. And since this cell, it is pluripotent, but it is not an embryonic stem cell, we call it induced pluripotent stem cell. So it's very similar to an embryonic stem cell, but it's derived from somatic cells. So now we have at our disposal the induced pluripotent stem cells, and we can use them to what we call the circle of promise. So the idea is to take somatic cells from a patient or a healthy subject. That can be skin fibroblasts, they can be blood cells, it can be keratinocytes. We know that we can reprogram all sorts of somatic cells. Doesn't really matter. The origin of the cell doesn't really matter. We can overexpress these factors I just told you about and derive the induced pluripotent stem cells. So then we can, in theory, take these cells and differentiate them back to the several somatic cell types. Now we have a cell that is very similar to an ES cell and has the same properties. For example, in the case of the lung, we can try to differentiate them to the different epithelial cell types, such as type 2 alveolar cells, the ones that secrete surfactant proteins, secretory cells, ciliated cells that are responsible for mucus removal. And then by having these cells now, we can try to do disease modeling, we can try to do drug development, and eventually, that's the long-term goal, to develop cell therapies. So how we could use these cells in the context of lung research? How can I derive lung epithelial cells from induced pluripotent stem cells or ES cells? And of course, as most scientists do, we go and look back at nature. So we get our lessons from nature. And the way to do, we know that from a blastocyst, we can derive cells from a blastocyst from the inner cell mass, become cells in the lung, and then these cells eventually give rise to the whole organ. And we know from developmental studies that the very first cells in the lung, what we call a lung primordium, express a protein known as TTF1 or NKX2-1. So you can see here a mouse embryo. We use a technique called in situ hybridization to detect the transcript of this gene. And you can see three domains of expression. So it's expressed in the lung primordium, the thyroid primordium, and the brain primordium. So it's not specific to the lung. And we thought that to study this primordium, we had to isolate the cells. The problem is TTF1 or KX2-1 is the transcription factor. So it's expressed, its localization in the cell is in the nucleus. It binds the DNA. And so if we want to isolate these cells and isolate them alive, because otherwise we have to kill them, use an antibody that's specific to NKX and then isolate them. So if we want to isolate live cells, we have to knock in what we call a reporter protein. So the idea is to take a reporter gene, for example, a fluorescent protein, and knock it into the locus of this gene. As each gene has two alleles, one will express NKX2-1 and the other one will express GFP. So when NKX is expressed, we will also see expression of GFP. And that makes our life much easier because then we can visualize the cells, we can look at the microscope and see green cells, and we know they express the protein we are interested in. So that's what we did. We created a knock-in embryonic stem cell line, and also we created a knock-in mouse. Using the same embryonic stem cells, we made a whole mouse that had the same, these cells, the NKX expressing cells had the same properties. And you can see here the lung of this mouse at several stages of development. So it's as early as embryonic day 9 days, 11, and you can start seeing the branching structure of the lung. Then E14.5, embryonic day 14.5, when that becomes more, the structure becomes finer, we have what we call branching morphogenesis that gives this fine structure that the lung has, the adult lung. And then right before birth, we see the very fine structure, where about the moment of birth, when the cells then will start becoming type 2 and type 1 cells. They give rise to the alveoli. So this is very impressive. In fact, the first time I saw that, I was screaming with joy because it took several years to develop this tool. And although these cells do not look as impressive, these are the more important cells. This is the lung primordial cells. These are the very first cells that take the decision in development to become lung, and then the whole organ can form. So if there is a defect, if the cells cannot take this decision or they cannot proliferate, the organ won't form. So using now the in vitro system, the embryonic stem cell line, we try to develop an in vitro protocol to differentiate these cells to lung epithelium. And we just published that 3 months ago. It was a back-to-back paper with J. Rajagopal's group from Mass General Hospital. So we published that in Cell Stem Cell, and it was very similar. Our work and their work reached very similar conclusions. And I'll show you in the next slide how this protocol goes. So we think of what we call directed differentiation. It's a very fancy way to say that, a multi-stage process of recapitulation of developmental milestones. But what it means, we have to look at development once again. We have to realize and understand which stages the cell has to go through to start with an embryonic pluripotent stem cell and end up being a differentiated cell, for example, a type 2 pneumocyte. And to do that, we get all our information from development. So people have done studies not only in mice, but also in fruit flies, in frogs, and now we have a lot of information that help us understand this process. And to make sure that we go down the right path at each stage, we have to monitor the cells to check for the presence of specific proteins before we proceed to the next stage. So we start with an embryonic stem cell, and we use REX1, it's just a marker. We go through several stages, and then we're supposed to reach the stage of the lung progenitor, and so we're supposed to see the expression of NKX2-1. So we did that, and we then look at these cells that were supposed to be lung progenitors. So you see now you will see a movie here. So these are cells we took at the anterior definitive endoderm stage. We put them in culture, and the next stage, in theory, is the derivation of NKX positive cells. So you will see, it's a 48-hour movie, but condensed into 11 seconds. We start what we call embryonic body. These are the structures that the cells form in suspension. We put them in a culture dish. Now cells start migrating out, and you will see that they also become lung progenitors. Okay. All right. Okay, so I don't see it here. So you can see here the cells. There is no green. So suddenly you see the cells appearing, the green cells migrating, proliferating. And so that's the moment. In fact, what we do now, it's the moment we simulate the moment of cell fate decision in vitro. So what we saw before as a lung primordium happens also in vivo, in vitro. Sorry. So we can take the cells now. These are the cells. You can see them here. We can take the cells, and then we can use a technique which is called flow cytometry. Since the cells fluoresce, we can separate it from the cells that do not express GFP. And we can replate the cells and study them in vitro. So there are several stages that follow this stage, because that's the very first stage in lung development. We have now the appearance of surfactant protein C, and everybody in this room knows that surfactant protein C is important for the function of the adult lung. But surfactant protein C appears very early in development, and there it doesn't have the function that it has in the adult lung. There it marks the early lung epithelia. So this is way before we have the appearance of type 2 cells that secrete the surfactant protein C. And it's as if all this was not complicated enough, we now have to go from this early lung epithelia to all the mature cell types in vitro. And that's very important because, and also very complicated, because we should be able in the future to direct the fate of these cells, the early lung epithelium, to derive type 1 cells, the ones that are responsible for gas transfer, type 2 cells, Clara cells, and ciliated cells. So this is going to be the big challenge for the future, how we can direct now these cells that are the early lung epithelium to all these different cell types. So going back to the circle of promise, we were happy now to have developed, because the first part of this, the derivation of iPS cells, have become a routine technology. We can reprogram any type of cell. And now we are happy to have also a protocol that we can use for differentiation of these cells. It's still early on. We cannot really derive any cell population in the pure form, but we know we are down the right path. So at that point, we started talking with Robin, and she said, why don't we reprogram? I have a lot of kids that have a disease that is due, a subset of child disease that is due to mutations in the NKX2-1 gene. So why don't I give you fibroblasts and you do what you know to do with all your other cells, reprogram them, get an iPS cell, and then try to do this, use these cells to model the disease in vitro. So what I alluded to in the first slide. And that was the central idea of the grant I wrote, that we can take these cells, reprogram them, and then use them for disease modeling. And the idea is that even though we reprogram these cells, they go through several stages. They revert back to an embryonic stem cell-like state, and then back to a lung epithelium stage. The DNA is damaged. There is a problem in the gene, in the NKX1 gene, and that's not going to change. So when we differentiate them back to a lung epithelial cell, it's going to be a cell that recapitulates the phenotype of the disease. So it's not going to function as a normal lung cell. And here you see we got the cells from the first patient, and this is the iPS colony. You can see here, stained with a marker of induced pluripotent stem cells and embryonic stem cells. So that was successful. We got several fibroblasts from 3 patients, and that's the first one. We did more detailed characterization, so you can see how we derive the cells. On the top, you see that we take the skin biopsy, we put the dermal fibroblasts, so we get the cells shipped to us from Robin. We put them in culture. Then we use our vector that has all the 4 reprogramming factors. We derive iPS colonies, and then we remove the cassette. So the sequence of all these factors, we remove it by using a technology that's called Krier combinase treatment. So these cells, we can tell that are also clinical grade because they do not have any exogenous sequences anymore. So we have removed the cassette. And then you can see on the bottom more detailed characterization of these cells. We use several markers to make sure that these cells are induced pluripotent cells. They have a normal karyotype, and we have also shown that we can derive additional disease-specific iPS from patients with SPC, SPB deficiency, SPC, SPB, and ABCA3 deficiency. And that's just to show you that these cells have been reprogrammed successfully. What you can see here is the presence of the exogenous vector in the genome of the cells. So that's before we remove it by treatment. So to go back to my first slide then, how we can use these cells. So once we have the cells, we have removed the transgene by treatment, and we have successfully reprogrammed these fibroblasts. We can now do disease modeling. We can try to develop drugs that improve the phenotype of the cells, that can reverse or mitigate the disease phenotype. And now, as you can see, that can happen, or it's happening through combination of in vivo and in vitro studies. So remember, in the first slide, on the left, we have developmental biology. And now if we look at that in the context of our system, we have development. We can study the developing lung by using a reporter protein. And on the right, we have what I alluded to as ES and iPS cell systems. So that in our case, it's the iPS we have derived from the fibroblasts, and is our mouse system here we can use to better refine our protocols that can then be applied into the human system. So it's something we start from the mouse, we develop our protocols in the mouse system because it's easier, and then we can apply them in the human system and derive now human lung epithelial cells and try to do what I just said, disease modeling, drug development, and also also derived functional transplantable lung epithelial cells. So that I want to conclude. I want to thank Darrell Cotton, my mentor. He's been a great mentor. Also, Jean and Amulia have been instrumental in deriving the cells, deriving the iPS cells. Robin and Seth Cole and Aaron Huffman. And last but not least, the Child Foundation, thank you for your support. Thank you for believing in us, and thank you for believing that what it seems to be science fiction today can become a clinical reality in the not so far future. Thank you.